Hi everybody, welcome back to Synthetic Biology 1. Today, we're going to talk about digesting DNA, cutting it up into smaller pieces using restriction enzymes. This is something we do all the time in the lab, but the exact protocol will change day to day depending on the application, so you have to be able to adapt. Working in the lab is a little bit like cooking. As a beginner, you have to follow the recipe exactly. But then gradually, you learn that some parts of the recipe can be changed, while other changes will ruin the food. One day you wake up and you're a master chef, and you can create your own recipes. Well, today, you will become a master chef of DNA digests. So let's start by looking at the ingredients of a typical digest reaction. The DNA is there to be digested. The main thing we need to know about it is its concentration. Typical DNA sample that we use in the lab might be 100 nanograms per microliter, but a wide range is possible. Your sample might be more or less concentrated by a factor of 10, and generally that's no problem. The enzymes are there to do the digesting. For these, we need to know the activity, which you can find in the documentation from the manufacturer. A typical activity might be 10 units per microliter, which means that 1 microliter of enzyme can digest about 10 micrograms of DNA in 1 hour. The reaction buffer creates the ideal conditions for the enzyme to act. The right pH, the right magnesium concentration, and so on. Wherever you buy your enzymes, they will always come with an appropriate reaction buffer. A common mistake is to think that reaction buffers are interchangeable, but in general they're not. So don't be a maverick, you follow the manufacturer's instructions. Water we add to control the total reaction volume. In this way, we can manipulate the concentration of all of the other ingredients. To set up a digest, we need to choose the right amounts for each of our 4 ingredients. Let's work backwards to see how to think about our choices. So the first question, what are the limits on your total reaction volume? Now, in general, I like big reactions, but you can't just fill your bathtub with DNA. There are limits on how big your digest can be. So for example, if you're planning to load your digest into a gel, then a typical well can only hold about 50 microliters. Or if you're going to load your digest into a DNA purification column, then these can only take about 300 microliters. If you go bigger than that, you have to consider that your Eppendorf tubes can only hold 1 or 2 mils. And then above that, it's like, what are you even trying to do, you crazy person? Why do you need that much DNA? The total reaction volume dictates the volume of the reaction buffer. These are almost always supplied at a 10x concentration. So that means in a 50 microliter reaction, we'll use 5 microliters of reaction buffer. The reaction volume also sets a limit on the amount of enzyme that you can use. So here's the thing. Reaction enzymes are supplied in 50% glycerol. Glycerol stabilizes the enzymes in the freezer, but it also interferes with the ability of the enzymes to cut effectively. As a rule of thumb, enzymes get messed up at about a 5% glycerol concentration. They just start cutting everywhere, non-specifically. It's called star activity. Your enzymes should be diluted by a factor of at least 10 into your final reaction. So for a 50 microliter reaction, we can only add a maximum of 5 microliters of enzymes. Now for me, I like to come in well under that just to be safe. So let's say 1 microliter. Nothing will ruin your day or your DNA like star activity. Now that we know how much of each enzyme we can add, we know the limits on our DNA concentration. In principle, 1 microliter of enzyme gives 10 units of activity, enough to digest 10 micrograms of DNA in 1 hour. I like to be very conservative here too and add much less DNA than the enzyme can handle. That way I'm covered just in case the enzyme is old, or maybe it's just not as amazing as the seller claims. But even 1 microgram of DNA is a ton of DNA for most applications. So for example, maybe you're just doing an analytical digest and you just want to see your DNA on a gel. Well, for this, 10 nanograms is plenty. If your goal is to purify the DNA and then use it in cloning, then you probably want a little more. But 1 microgram of DNA is enough for 99% of what you'll want to do in a lab on a normal day. Now we built our recipe. We'll put our samples in a 37-degree incubator and we'll leave them there for an hour or more. But how long exactly? Well, we still have one more important question. How important is it that your reaction goes to completion? Do you really need to cut up every last piece of DNA, or is a little bit of uncut stuff okay? You see, here's the thing. The first 99% of the DNA is easy for the enzymes to find and cut. But as the concentration of uncut DNA gets lower and lower, the reaction rate slows down. Those last few pieces of DNA take a long time to get. Now, if you're just running a gel, then you don't care. You won't see those 1% of the fragments anyway. But if you're going to use your sample for a PCR, then sure, you can easily amplify those undigested fragments. If you're going to transform your sample, then you really need to be sure to get those last few pieces, because they will easily transform into cells and possibly contaminate your downstream reactions. Okay? So that means using much less DNA than the enzyme can handle and letting the reaction incubate for longer, 2, 3, or maybe even 4 hours, just to get everything. And voila, you are now a master chef of DNA digests. With a little practice, your digests will come out delicious. Just kidding. You should never eat in the lab.
Thank you.